I'm Daria Zikaus, and I'll tell you about our paper entitled Cell Division in Tissues Enables Macrophage Infiltration. My lab studies tissue invasion, a process in which a cell enters and moves through an environment of densely packed surrounding cells. This capability is essential. In the embryo, macrophages invade tissues to help organs form and then stay to affect homeostasis even in the adult. Throughout life, additional immune cells must enter tissues to control infections. And if cancer forms, immune cells respond by infiltrating the tumor to try to control it. If they don't manage, metastatic cancer cells can then invade surrounding tissues and ultimately distant organs. But what determines when such tissue invasion occurs? Is this governed just by the efforts of the migrating cell, or can the tissue itself act as a gatekeeper? We have now discovered that surrounding cell dynamics can control tissue invasion using the elegantly simple in vivo model of macrophages disseminating in the Drosophila embryo. Embryonic macrophages migrate to a place where they then infiltrate between two closely opposed tissues, the ectoderm and the mesoderm. The most challenging step is the penetration of the very first macrophage, which must make a path for itself and for the macrophages behind it. My postdoc, Masha Akhmanova, observed that the first macrophage always entered between these two tissues when there was at least one ectodermal cell dividing next to the site of invasion. To test the importance of this, Masha genetically modulated divisions just in this flanking ectodermal tissue. She found that if no neighboring cell was dividing, then no macrophages entered. She even showed that more ectodermal division caused more macrophages to enter. Thus, division is a rate-limiting step for invasion and somehow functions to enable it. But what is a dividing cell doing that allows macrophages to invade? Through live imaging, Masha observed that invasion could occur already in the first phase of division, so-called mitotic rounding. During this time, a cell increases its tension, becoming stiff and round, and thereby opening more space around itself. Then this rounded cell disassembles the adhesion sites that attach it to its neighbors. Masha tested which of these processes is required for invasion. First, with help from the Durensis lab, she used optogenetics to artificially increase the surface tension of ectodermal cells at and near the entry site. This led the ectodermal cells to round up and thereby increase surrounding extracellular space, even though they were not really dividing. But these changes were not enough to allow macrophages to enter before the true division process occurred. However, when Masha reduced focal adhesions by genetic knockdown, she found that macrophages were able to invade independently of division. This proved that the key step that occurs during cell division is the disassembly of the focal adhesions which attach the ectoderm to the underlying ECM and mesoderm. Thus, our work provides a simple and powerful new perspective on invasion into closely packed cellular environments. We show that the dynamics of the tissue itself can control cellular entry. Factors that alter tissue cell division could thus also affect the number and location of immune cells that enter during development or inflammation, and potentially even of cancer cells during metastasis. We are very excited to explore these implications for vertebrate and human biology and disease. The driving force behind this work, as you have probably noticed, was my wonderful postdoc, the first author and co-corresponding author, Masha Akhmanova. She was actually trained in biophysics, but she fell in love with microscopy and genetics during this project. She was aided by several other members of my lab, Shamsi Imtinani, Attila Gyurgi, and Mariana Guarda, and she built on the foundation laid by my former postdoc, Aparna Ratish, who is now a professor at the University of Warwick. Masha also had help from three local high school students, Mikhail Vlasov, Fedor Vlasov, and Andrea Kopian, who worked on algorithms and fly genetics. And finally, Masha conducted her optogenetic experiments with expert guidance from Daniel Kruger during a visit to Stefano Durentis' lab at EMBL, Heidelberg.